Hi guys, welcome to all members of Botany for You. Myself Rahul Jawale, research student from Government Vidarbha Institute of Science and Humanities, Amravati. Today we are gathered here for the national workshop on CSIR-UGC-NET-JRF preparation in life science. I am thankful all the founder member of Botany for You and all respected teachers and students from various universities. Today I am going to deliver a lecture on gene silencing. Before going toward the gene silencing, we must have to understand the mechanism of protein synthesis. You've seen the diagrammatic representation of protein synthesis, and you know very well there are two stages in protein synthesis: transcription and translation. During translation, mRNA is formed, and during translation, protein is formed. Okay, so these are the two stages of protein synthesis. So what exactly happened in the gene silencing? These two stages become blocks. First block at the transcription level and second block at the translation level. Okay. Then what is the need of gene silencing? If gene is mutated, then the expression is also mutated. In that particular situation, cell decide to silence the gene, or we can apply the gene silencing technique. Or if in case viral particle attack the cell, in that particular situation, cell also follow the gene silencing mechanism. So now come to the point, what is gene silencing? Gene silencing is a technique used to reduce or block the expression of corresponding gene. If unwanted gene is present inside a cell, in that particular situation, cell decide to block the expression of that corresponding gene. Okay. So gene silencing is generally described as switching off the gene by a mechanism other than genetic modification. Okay. And gene silencing occurs when RNA unable to make a protein during translation. Or we can say gene silencing is same as gene knockdown but totally different from the gene knockout. On the next slide, we see the different ways of gene silencing. That is gene knockout, gene knockdown, silencing of gene by using RNA technology, in which antisense RNA technology and RNAi technology, that is RNA interference technology. So these are the some ways of to silence the gene. So what exactly happened in the gene knockout? In which the complete portion of the particular or corresponding gene removed. Okay. But actually gene knockout is not a part of gene silencing because it includes recombination techniques. What? Recombination techniques. So second one is the gene knockdown. It is actually follows the gene silencing mechanism. And antisense RNA technology and RNA interference technology plays very important role in the gene silencing. So next, what to do for the gene silencing? Gene can be regulated at transcription level or post-transcriptional level. As we seen in the diagrammatic representation, in which we see the protein synthesis and we block the stages of that particular protein synthesis process. Okay. So similarly, we can regulate the gene expression at the transcriptional level or post-transcriptional level. So there are mainly two types of gene silencing: transcriptional gene silencing and post-transcriptional gene silencing. So next, what are the transcriptional gene silencing and post-transcriptional gene silencing? So transcriptional gene silencing are the genomic imprinting, paramutation, transposon silencing, transgene silencing, position effect, RNA directed DNA methylation. So all these are the transcriptional gene silencing. But we seen this all the transcriptional gene silencing in the next lecture. Now we are only focused on the post-transcriptional gene silencing, that is antisense RNA technology and RNAi technology. In RNAi technology, there are three different types of RNA, that is double-stranded RNAs, that is mRNA, shRNA and siRNA. So what is miRNA? That is the microRNA. Sh is the short hairpin RNA and Si is the small interference RNA. Okay. So stepwise we see in the detailed mechanism of antisense RNA technology and RNAi technology. Now next, that is the post-transcriptional gene silencing. So what is post-transcriptional gene silencing? The ability of exogenous or sometimes endogenous RNA to suppress the expression of the gene which corresponds to mRNA sequence. What does it means? It means if we want to suppress the expression of particular gene, we have to broken down the mRNA sequence or we block to mRNA sequence. So how do we block mRNA sequence? We block mRNA sequence exogenously or cell itself block, that is endogenously. Now we are going towards the antisense RNA technology. So what is antisense RNA technology? The name itself indicate the antisense. Okay, it means it contains the complementary sequence of the particular gene or particular mRNA. Okay, so antisense RNA contain opposite complementary sequences of mRNA. If antisense RNA and mRNA present in same cell, it leads to formation of stable duplex. Duplex interfere with gene expression, while this technology widely used in the plants. Now consider there are two cells. Okay, one is normal cell and second one is antisense RNA technology cell. So in normal cell what happens? mRNA is formed from the DNA or particular segment of DNA, that is gene, by the process of transcription, and that mRNA is translated in the form of proteins. This is the normal process. But what we actually do in the antisense RNA technology, we copied the mRNA sequence. We copied the mRNA sequence artificially. Then that antisense RNA inserted inside the cell cytoplasm or inserted inside the organism, then that antisense RNA recognizes the complementary sequence of that particular mRNA and form a stable duplex. Clear? When mRNA and antisense RNA form a duplex at that particular point, we can observe there is no further expression of that mRNA. It means translation inhibited. Okay. So what next? If duplex is formed at that particular point, RNase H is recruited on the duplex, and that RNase H particularly break the mRNA sequences. If the mRNA sequences are broken down, then the expression of that particular mRNA is inhibited. So ultimate aim of the gene silencing is completed here. Okay. Now we seen the RNA interference technology. What is RNA interference technology? It is a process in which double-stranded RNA silence gene expression by degrading mRNA or inhibiting the expression of that particular mRNA. It means inhibit the translation. In RNA interference technology, first thing is important, that is need of double-stranded RNA. Secondly, it is selectively gene knockdown. As we seen in the second slide, I told you these all techniques are the gene knockdown, not a gene knockout. Okay, clear? Then RNA interference operate its natural role for virus defense and endogenous gene regulation in plants. As I told you, if the gene is mutated or if the cell is attacked by the virus or viral particle, in that particular situation, the plant cell endogenously perform that RNAi technology. Okay, so now consider similar situation. There are two cells, one normal cell and one cell in which we can apply the antisense RNA technology. So in normal cell, what happens? DNA is transcribed into the mRNA and mRNA translated into the protein. This is the normal process. So what we do in that process? We inhibit the expression of that particular mRNA. So in the next cell, consider that there is a nucleus. Inside the nucleus there is a DNA, and DNA is transcribed in the form of mRNA. That mRNA come inside the cytoplasm and translate it into the proteins. But actually we want to inhibit that processes. Okay, we are seeing the post-transcriptional inhibition. So in that particular situation, we have to inhibit the post-transcriptional stage, that is the mRNA stage. So we have to inhibit the mRNA by using RNA interference technology. In that RNA interference technology, there are number of factors like dicer, drosha, and other some factors are used to break down or block the expression of that particular mRNA. Clear? Next, overview of RNA interference. First one is double-stranded RNA cut into short double-stranded RNA, that is small interference RNA by enzyme called dicer. As I told you, RNA interference technology needs double-stranded RNA. But that double-stranded RNA is processed by using the enzyme called dicer. That enzyme broken down that double-stranded RNA. Okay. Then this RNA base pair with the mRNA present inside the cell cytoplasm, while with the help of RISC complex, degradation of mRNA takes place. It is highly specific process. Okay. Then it is very potent activity. It shows very potent activity usually seen in the eukaryotes. It actually seen in the prokaryotes, but majorly seen in the eukaryotes. Near about 30% of genome is regulated by RNA interference technology. Then there are three types of double-stranded RNA used in the RNA interference technology or RNA interference pathway. First one is the small interference RNA, that is siRNA. Second one is the microRNA, that is miRNA. And third one is the short hairpin RNA, or we can say it is shRNA. Clear? So next, what are the main components of RNA interference technology? So first one is the drosha, second one is the dicer, third one is RNA induced silencing complex, that is the RISC complex, then argonaut, and last one is the RNA dependent RNA polymerase. So what drosha actually do inside a cell? It is actually work in the miRNA technology, that is the microRNA technology. Drosha actually process the pri-miRNA to pre-miRNA. Second one is the dicer. It is a RNA's three like double-stranded RNA specific nucleus, or we can say ATP dependent nucleus found in the C. elegans, which actually cleaves that double-stranded RNA. Okay. Next one is RNA induced silencing complex, that is the RISC complex. RISC complex is the complex made up of 2 to 3 different subunits, including argonaut, slicer. Then what is argonaut? Argonaut is a protein molecule made up of near about 110 to 130 amino acid protein. It is a part of RISC complex. Now last one is the RNA dependent RNA polymerase. It is used for amplification of double-stranded RNA. Now, if we're talking about the dicer contains different domains like helicase domain, like PAS domain, like RNase 3 activity domain. There are two RNase 3 domain present inside the dicer, and double-stranded ribosome binding domain present inside the dicer. Now we seen the first type of double-stranded RNA in the RNA interference technology, that is small interference or RNA, or we can say siRNA. siRNA is 21 to 23 nucleotides double-stranded RNA mediates post-transcriptional gene silencing, that is PTGS, post-transcriptional gene silencing. siRNA produced inside a cell, it means in vivo by cleavage of double-stranded RNA, while it is incorporated into the RISC guiding to mRNA. When that double-stranded RNA is processed by the dicer, after that it is incorporated into the RISC complex to guide the mRNA. Each strand of siRNA has 5' monophosphate termini and 3' hydroxy termini. There are 2 to 3 nucleotides overhanging at the 3' end. When dicer process the double-stranded RNA at that time, 2 to 3 nucleotides overhanging sequences form at the 3 termini. siRNA is complementary to specific sequence of target mRNA for degradation. Amplification of that RNA is held by RNA-dependent RNA polymerase, that is RDRP. Next, now we see in the mechanism of siRNA, or we can say small interference RNA. Firstly, we focus on the points. First point is double-stranded RNA chop into the siRNA by dicer. Second, siRNA recruit RISC complex. It unwind the siRNA. Then third, unwound siRNA base pair with complementary mRNA. Then gRNA, that is the guide RNA, identify target mRNA. Next, target mRNA cleaved and degrade. So these are the points. Now we focus on the diagrammatic representation in which you clear all the phenomena. This process can be happen endogenously or exogenously. If double-stranded RNA is present inside a cell, so that double-stranded RNA is processed by using the enzyme that is dicer. But when dicer process the double-stranded RNA, when double-stranded RNA present inside the nucleus, and that double-stranded RNA come out from nucleus to the cytoplasm with the help of importin and exportin proteins. Okay, this is the mechanism for the transporting of molecules inside the nucleus and cytoplasm. So when the double-stranded RNA come inside the cell cytoplasm, at that particular time, dicer cleave that double-stranded RNA and form a siRNA. You've seen in the diagram, there are two short stretches of the siRNA present inside a cell. Dicer is perform the RNase 3 activity. So as I already told you, RNase 3 having endonuclease activity and it cuts double-stranded RNA. Okay. So when the siRNA is formed, that siRNA is processed by the RISC assembly. So what is the RISC assembly? So RISC assembly is actually made up of the Argonaut and slicer. As I already told you, RISC is the RNA induced silencing complex. So that RISC assembly bound on that siRNA and processed it. How it processed it? So Argonaut present in the RISC complex perform the RNase H activity, and RNase H having activity like it cuts single strand, and that whole RISC assembly unwound the double-stranded siRNA. When the double-stranded siRNA unwound, at that particular time, one strand act as a passenger strand and another strand act as a guide strand. When helicase activity done by the RISC assembly, at that time, the passenger strand and guide strand separate. Passenger strand leaves that assembly and guide strand remain there in the RISC complex. Now the assembly is RISC complex with guide strand inside the cell cytoplasm. Now guide RNA having complementary sequences of that particular target mRNA. It recognizes the mRNA and bind with the complementary sequence of that mRNA. When the guide RNA recognizes the mRNA sequences, it forms the complex with the mRNA. So we can call it mRNA guide RNA complex with the RISC assembly. So what next? RISC assembly starts to perform their activity. Now Argonaut and slicer present in the RISC assembly start to break that mRNA into the small pieces. We can see cleavage of mRNA. Then that cleave mRNA unable to form a protein. We can say we terminate the translation process. Then that cleaved pieces of mRNA start to degrade. It means here the ultimate aim of gene silencing is completed with the help of siRNA technology. Now come to the second RNA interference technology, that is microRNA, or we can say miRNA technology. In miRNA, there is a single stranded RNA molecule. Actually, it contains a complementary sequences and then it forms a double stranded molecule. It is near about 22 to 25 nucleotides. The microRNA is encoded by gene but not translated into proteins because it is a non-coding RNA. Next, primary transcript pre-miRNA is processed into short structure called pre-miRNA and finally into functional miRNA. Now come to the diagrammatic representation. Imagine there is a single cell and inside the single cell there is a nucleus. Okay. Inside a nucleus you can see the structure of DNA, and from DNA there is a one transcript is formed with the help of transcription. That double-stranded transcript is called as pre-miRNA. That primary transcript is processed by DROSA and DGCR8. That DROSA and DGCR8 processed pre-miRNA and converted it into the pre-miRNA inside the nucleus. Now that pre-miRNA transported from nucleus to the cytoplasm with the help of exportin and RAN GTP through the nuclear pore complex. When the pre-miRNA come inside the cytoplasm with the help of exportin through nuclear pore complex, the dicer is there. Dicer perform the RNA's 3 activity, endonuclease activity. It performs the double-stranded cut and finally form the mature miRNA. You can see clearly inside the diagram. Now mature miRNA is ready to block the expression of that particular targeted mRNA. Now the further mechanism is similar with the siRNA technology. Now what happens? RIS complex is come towards the mature miRNA and make an assembly. In RIS complex, what happens? There is an Argonaut and slicer. They break bond between the two strands and one strand from this act as a passenger strand and another strand act as a guide strand. So what happens? Passenger strand leaves that complex and guide strand remain there. So now the complex is with the guide strand. So now RIS complex with guide strand goes toward the targeted mRNA. The guide strand recognizes the complementary sequences of that particular mRNA and make a complex. Targeted mRNA guide RNA complex. When the complex is formed, the RIS complex process the targeted mRNA. It means it break down the mRNA. When the mRNA is broken down, it means ultimate aim of the gene silencing is completed, because when the mRNA is broken down, it unable to translate into the proteins. Now that broken down mRNA is degraded, it means the short segments of that mRNA are further degradation happens. Now coming to the next RNA interference technology, that is short hairpin RNA. Short hairpin RNA technology is similar with the siRNA technology, but there is a small difference. In hairpin model, it forms a small hairpin loop. That's why the name is hairpin RNA. You can observe inside the diagrammatic representation, there is a nucleus, and inside the nucleus there is a double-stranded RNA. That double-stranded RNA is formed from single-stranded RNA. That single-stranded RNA having a complementary sequences. That complementary sequences bound together and form a double-stranded RNA. Now that double-stranded RNA come out from nucleus to the cytoplasm. That double-stranded RNA near about 20 to 25 nucleotides. When it comes out into the cytoplasm, it is processed by dicer. Dicer perform the RNA's 3 endonuclease activity and form the double-stranded cut. When double-stranded cut is formed, the 5' monophosphate is there and 3' hydroxyl termini. At the 3' hydroxyl termini, 2 to 3 nucleotides are overhanging. That processed double-stranded RNA now 15 to 20 nucleotides because dicer cleave that double-stranded RNA and at that particular time, small portion of that RNA cleaved out. That's why firstly 20 to 25 nucleotides double-stranded RNA, now 15 to 20 nucleotides. Now, processed double-stranded RNA is called shRNA, or we can say short hairpin RNA. Now, further mechanism is similar with the siRNA or miRNA. This complex come toward the double-stranded RNA, that is shRNA, and performs the helicase activity. At that time, one strand acts as a passenger strand and another strand acts as a guide strand. Passenger strand leaves the assembly and guide strand remain there. Guide strand with the RISC complex go toward the mRNA. In RISC complex, Argonaut and slicer play important role to unwound these two strands, and Argonaut perform the RNase H activity. RNase H having capacity or capability to break single strand. Now the RISC complex with the guide strand go toward the targeted mRNA. Guide strand found the complementary sequences of the mRNA and make a complex. Now the targeted mRNA is processed by RISC assembly and broken down into the small fragments. The small fragments further degradation takes place. Now here ultimate aim of gene silencing done. Now next, advantages of RNA interference. Downregulation of gene expression simplifies knockout analysis. RNA interference technology is easier than antisense technology. When we perform the RNA in which siRNA more effective and sensitive at lower concentration. RNA are used to block expression of unwanted gene. It is helpful to induce the viral resistance. It is used to analyze unknown gene in sequence genome. While it is useful approach in future gene therapy. As we seen the advantages of RNA interference technology, likewise there are some disadvantages of RNA interference technology, like high pressure injection and electroporation can cause significant injection damage to the normal tissue and organ. If you used the electroporation technology or high pressure injection, it may cause or damage the tissue or organ. Then the liposome encapsulated siRNA may also be toxic to the host and may cause severe host immune responses. So these are the disadvantages of RNA interference technology. In this lecture, we had seen the post-transcriptional gene silencing. But there are certain factors in the RNA interference which we cannot cover here. So in the next lecture, we going through the overall process of gene silencing, in which we seen the transcriptional as well as post transcriptional gene silencing in detail. Okay, thank you.